afternoon, everyone. Thank you for giving me this opportunity to introduce our research today. Today, I will talk about our recently published paper titled Plasmon Enhanced Single Extracellular Vesicle Analysis for Cholangiocarcinoma. Before we start today, I will briefly introduce our center. The Center for Systems Biology in MGH is focusing on multidisciplinary research, combining medicine, biology, and engineering. So there are internationally eminent scholars in with different background and expertise. So whenever you come up with some ideas, you can find the person you need in the center. So it's very collaborative and interactive. In addition, if you have some idea and have some technology, you can apply this technology to patients because in MGs there is some network between clinicians. So if you are looking for an institute which you want to apply your knowledge to the disease with other field people, then I think it would be a good choice to apply here. In my case, my major is toxicology and pulmonary system. So we are in another project that I am working with pulmonologists and thoracic surgeon for analyzing the data and collecting the samples. And now we start now. Cholangiocarcinoma (CCA) is one of the fatal disease, which 5-year survival rate is under 20%. Actually, it has a biomarker CA19-9, but its accuracy is not that good. Sometimes it is elevated because of the obstructive jaundice. So many researchers are trying to get the better biomarkers. So some people are using bile because the CCA secrete their cytokines or proteins to bile. So they're using bile for discovering biomarkers. However, there is some limitation for the research of the bile. So more sensitive ways are needed for biomarker, and the biomarkers discovery is still ongoing. Actually, cell can secrete various biomolecules such as proteins and nucleic acids and extracellular vesicles. Especially extracellular vesicles actually play a role of a surrogate of cells because it can contain the various biomolecules like the parent cells. So there are many. So it's a kind of a very good virtue of the biomarker. So many researchers have published EV-associated research demonstrating that EVs can be good markers in various diseases. So there are very fundamental two papers. The left one is demonstrating that EVs can transfer their parental cells and nucleic acid to other cells. It inferred that EVs can propagate parental cells properties to recipient cells. It's a kind of a functional role of EVs. And the right paper, you can see that very similar pattern between exosome and cells biomarkers. So it means that exosome can be a biomarker of cells by mimicking the cell's properties. So we premise that EVs can be used used as a biomarker in our CCA project. However, we usually in user research and EVs, we are dealing with some bulk EV analysis. Even though you isolated EVs from biofluid, we cannot say they are derived from tumor. They are secreted from not only tumor cells, but also from normal cells. So in EV population, it has very heterogeneous EVs. So even so, in the case, if in the early stage of a tumor, you only have a very small amount of tumor-derived EVs. So we need some if we want to detect some early stage of tumor, we need to detect very sensitively EVs by one. So this study is to visualize a single EV and detect cancer-derived EV. For this job, we should enhance the signal of EVs and set up the analysis of EVs methodology. In our lab, we developed the flex chip, which is the nanoparent wafers. The periodicity is 500 nanometer. Actually, on the edge of the hole, there is a plasmonic resonance. Actually, this resonance induces the enhancement of the fluorescence. So as you can see that AF555 and AF647 can be enhanced on this chip. To be specific, we compare plain gold and the nanoparent gold flex chip. So after labeling EVs with a fluorophore, we attach these EVs on the substrate. Then we followed by immunolabeling. Then you can see that EVs can be observed more, and the average EV intensity increased about 18 times more. So now we can see, then this system could increase the sensitivity. And for choosing which fluorescence would be best for our system, we label the EVs with different colors. So we labeled with EVs for A555, 647, and size 7 fluorophore. And you can see that AF555 and the 647 show the more EV count and higher intensity compared to plain gold. So we decided to use AF555 and AF647 channels for our platform. Then we confirmed that our system can be used for sensitive system. Then we needed to decide which markers we are gonna evaluate. So we cultured 4 types of cholangiocarcinoma cell line. The first one is the normal cholangiocyte cell line, and the other three are CCA cell lines. So we analyzed the proteomics data of cells and EVs, and you can see that AbCam, EGFR, and MARC1 show the increased value compared to normal cell lines. Actually, we got more markers in the proteomics, but after validating with flow cytometry, we decided to AbCam and EGFR MARC1 would be the best combination. And after that, we confirmed these markers expression on the tissues as well. Then we decide which system will be used and which fluorescence will be used and which marker will be evaluated. Then I will explain how we evaluate the intensity of the markers. Actually, every image we have two channels. EVs were labeled with AF555, and the markers were labeled with AF647. So there are two channels, and after we evaluate the co-localized markers intensity here. So we got the location information of EVs from our system, and then we calculate the intensity of the marker on the 3x3 pixels of the EV's location. For evaluating the marker's expression, every time we measure that, we conducted experiments with isotype control. So we set the threshold for the marker. So after we setting up the threshold, we calculate how many EVs are positive EVs and calculate the average of the intensities as well. So with this method, we evaluated these three types of biomarkers here. So to validate our method and our system is working well, we compare our method with the conventional method together. So bead-based EV flow cytometry is one of the bulk EV analysis. So as you can see that these two methods are correlating with each other. On the other hand, Flex show the better limit of detection. Actually, it is over 10,000 times better than bulk analysis. So when we compare to other bulk EV analysis, our LD value was pretty much better. So we moved on to clinical samples to see how these markers and how these methods are well going with the clinical application. So in the ERCP procedure, the clinicians collected the biopsies, and after several differential centrifugation, we collect EVs with three types of methods. This is because there is no standard method with bio-EV. So we tested the ultracentrifuge, the first method. The second is a side exclusion chromatography, and third one is a combined method. So as you can see that for the when it comes to protein concentration, UC and SEC show the higher values compared to UC-SEC. But in the number of particle, when we use the ultracentrifugation, we show the lower than side exclusion chromatography. This is because ultracentrifugation uses a very high gravity, so it can easily aggregate particles together. So that's that as you can see, the protein amount per EV number in UC groups shows the highest value among these three types of methods. For the UC-SEC, actually it has a too low yield of isolation compared to UC and SEC. So we decided to isolate bile EVs using SCC. So this is the FLEX data with the bioclinical samples. So you can see that ABCAM, MUC1, and EGFR were evaluated from 17 cholangiocarcinoma patients and 8 benign patients. So you can see that MUC1 showed a very significant distinguishable value between benign and cancer patients. And when we combine these three kinds of markers together, you can see that much better p-value and better AUC in ROC curve. This is because there are several patients whose MUC1 MUC1 expression was very low, but in these patients show the higher ABCAM and EGFR expression in the FLEX signal. So that's the reason when we combine the intensities of these three markers, they show the 100% sensitivity and much better accuracy than MUC1 when we consider only MUC1. So we concluded that FLEX chip can show the very good sensitivity with the multiple channels, including AF555, 647, and Cy7. And the diagnostic markers, MUC1, ABCAM, and EGFR that we discovered, actually they show the very good AUC values and accuracy. For the next step, maybe we can expand to mutated oncoprotein, because in the early stage, the mutated oncoprotein could be existing in the blood EVs as well. And if we could use a multiplexed system, we could immunolabel EVs with different colors, and we could make some subpopulation of the EVs. And actually, ERCP is the kind of burden to clinicians and patients. So if we discover the organ or CCA specific markers in the blood, then we can apply these markers to detect CCA EVs in the blood. And our combined biomarkers can be used in the longitudinal study, like before surgery or after surgery, or to keep watching the prognosis in the future study. These are the authors in this study. Actually, Taehwang has worked on optical optimization, and he's doing some biological things as well. Yugyang, in the case of Yugyang, she's working on validating proteomics data and preparing some clinical samples. Youngsun is my PI in our lab, and he has given comments to me about FLEX chips and EV's characteristics. And CJ Ralph and Junyan Min has been giving us comments weekly meetings. So thanks to these people, everyone. This study is funded by NIH and Ministry of Trade, NRF, and Yonsei University. And I really want to appreciate to everyone in our center who create the great environment for research. This is the end of my presentation. Any questions are welcomed.